Roberto Colter is Professor Emeritus in the Department of Microbiology and Immunobiology at the Harvard Medical School and is also Director of the Microbial Sciences Initiative at Harvard. He joined the Harvard Medical School in 1983, where he has conducted extensive research on microbes ranging from basic bacterial physiology to bioactive compound discovery. He's published extensively on his research and has received many awards and honors for his contributions to science, as you might imagine. But he's also, and very importantly for those of us in my business, deeply committed to education and has to date trained over 120 individuals who have pursued careers in science in both academic and industry settings. And he also shares his expertise through workshops and presentations for the general public and budding microbiologists. He started Harvard's Microbial Sciences Initiative, which is an interdisciplinary program that brings together scientists from across the university to work on projects that advance a comprehensive understanding of the microbial world. Scott Chmielewski is a microbiologist, science photographer, and author based in Roberto's lab. He specializes in the development of imaging, photography, and scanning techniques and technologies aimed at understanding microbial multicellular forms, collective behaviors, communities, and interspecies interactions. Luckily for us, he is also interested in leveraging the power of photography as a medium for communicating microbiology to other scientists and to the general public. And his work has been featured in many scientific journals, as well as publications such as Wired, Time, The Atlantic, NPR, Scientific American, and Smithsonian Magazine. And you'll be able to actually admire some of his amazing photographs in the exhibit upstairs. Roberto and Scott have collaborated on many projects, including our exhibit, and last year they published a beautiful book called Life at the Edge of Sight, a photographic exploration of the microbial world. And you will actually be able to see copies in the museum store upstairs, which is open and ready for business while you're up there. Just have to put a little plug. All funds go to a good cause. Anyway, Roberto and Scott, they make a dynamic duo. They've been a huge pleasure for us to work with on the exhibit, and I am so looking forward to hearing what they have to say as they share their passion for the microbial world. Thank you.
Thank you, Jane. Thank you very much. I should say the first person I really need to thank here is Jane. About 3 years ago, we walked into her office and proposed a microbial life exhibit at the Museum of Natural History here. And I don't think it took too much convincing. I thought you liked the idea, and we've worked at planning this, and this is a moment of great joy for us, and I'm very happy that you all came to share in this very special moment in which we get to talk a little bit about the exhibit, and then hopefully many of you will walk up there, enjoy some microbially produced foods along with beverages, and also see the exhibit, which I think has really turned out beautifully. So before I get into the subject matter, I'd like to give the thanks, because I think that's quite important. This has been a project that's involved a lot of people, and I want to make sure that credit doesn't go unnoticed by leaving it at the end. So first, the major funders for this in terms of organizations, I have to thank the Sloan Foundation. And Paula Olszewski is here, and she was the director of the microbiology of the built environment, in which I was a member of the advisory board. And really a lot of the exhibit that you will see, and I'll talk about it here, is inspired a lot about the research and the development that has happened in the last 10 years, in which we've had people really deeply investigating the microbiomes, the microbiology of the built environment. I think that's important. NSF, also funded, and this Microbial Science Initiative that I think I got too much credit. A lot of us together, and two of the co-founders are here, Rich Losick and Colleen Cavanaugh. 15 years ago, we decided to have breakfast and talk about science, and it's still going on, the same thing. It's really a dynamic initiative in the campus. It's something that I really love. So now let me just mention a few people who have been very, very important in this. Aside from Jane, and I've really enjoyed working with the whole team at the exhibitions department, but I have to mention that it's been a joy to work with Jan and with Sylvie And Jenny always is smiling. So I really am inspired by, no matter how the pressure comes, Jenny smiles. And Tristan, what a person to come through with all of the work. Wonderful, wonderful. And the whole team at Exhibitions has been absolutely fabulous. And education, I think this is really a neat aspect of this exhibit. We're going to be having volunteers that are, some of them are actually microbiologists who really want to get involved in science communication to the public. And there's going to be demos, live demos, a little bit of what we're going to be showing today, live demos so that people can really interact with the microbes and with volunteers to learn the science. And Wendy leads that department. And I have to say that we have in the range of almost 40 volunteers that have been training so that this thing can be going on for the length of the exhibit. The Sloan Foundation, I already mentioned, Paula Olszewski was instrumental in providing some of the funding. And we had an advisory board that included Colleen Cavanaugh and Peter Gergis and Andy Knoll. And most important, we have very special donors that care about communicating science to the public. So Clark and Susanna Bernard, thank you very much. Much appreciated. Very important. Very important. Now, all of these people and the project itself stand really on the shoulders of a major giant. And when he comes on stage, you'll see why I say giant. But he's a giant, and I have to say that not enough credit can be given to Scott, how much he inspired this. He's been working on it very hard, and he's been the force behind it. So Scott, thank you very much. So that's the thanks. So I'm really excited. I'm nervous. You know, they say there's a few talks that you only give once in your life, like your thesis defense. I didn't wear a tie for that. I think this is the only tie that I own. I was very nervous, much as my wife knows. I didn't know what to put on. So I said, why not, you know, use it after 25 years? You've never used a tie. This is really special. So I'm very nervous. I feel my pulse. And it must be like, you know, this is like more than when I run every morning. It's really, can you guys feel your pulse? Are you guys as excited as me? Can you feel your pulse? It's a good sign because that means you're alive, right? So it's good that you can sense your pulse. So can you feel your pulse? Well, you know, something magical happened last November in that NASA released what I consider one of the most remarkable videos that I've ever seen. And the reason I find this is because for the first time in the history of humanity, in the history of this planet, you can think that all this wonderful biosphere and all the evolution that has happened has reached the point that it's been able to take a selfie. That is, humans sent satellites up there, and for the last 20 years have been monitoring what has happened to this planet. And just like us, that we just sense our pulse, the planet has a very vital sign, which I like to call the Earth's pulse. And let me just talk about this as you watch it, because it really is, just imagine all that it took to make this video. And the point for showing you this video about the vitality of this planet is that when you think about it, when you consider what's happening, it's really foundational, the activity of the microbes. And I'm going to make an exaggeration, and I'm going to focus first on the green areas that are land. And yes, that's, of course, the action of plants. But if we think about plants, and we think about their origin, their evolutionary origin, it's that sometime, a long, long time ago, one cell swallowed another one that was a photosynthetic bacterium, and it became the chloroplast. And so you can argue that the foundation of all of the plant activity is very much based on what was originally a microbe. But if we go to the oceans, and watch these activities, what we're seeing in these lighter blue areas are blooms of photosynthetic organisms, be them phytoplankton made of algae or be them cyanobacteria. And they are really keeping the planet going. They're producing the oxygen that we breathe. And they're even involved in the melting and the forming of the ice that we see going back and forth. So really, this planet is a microbial planet. Now, we have, of course, come in and began to affect it. But I think it's important to recognize how the planet, through its microbial activities, is sustaining life like ours. And that's quite important because we stand now in a moment in history that this vital sign, this pulse, could begin to be tampered with. And something to think about how we maintain the planet and how we maintain its pulse, because it really is an honor to the microbes that have been doing that. But of course, when I thought about giving tonight's lecture, I had to say, well, this is a lovely way to start, but come on, you've got to bring it down to earth. Right, so bring it down to earth. You know, this I can talk about satellites and wonderful things, but let's bring the microbes down to earth, to something that we really can connect because it's something part of our daily lives. And that's why I like so much the microbiology of the built environment. So we know a lot of microbial environments all over the world, and yet until 10 years ago, we really hadn't studied one of the most important environments, which was those environments that we have built and where we spent somewhere between 80 and 90% of our time. Like this room. You know, what's going on in the microbiology in this room? And so I decided that there were very nice center, and you'll see it reflected in the design of the exhibition, is to go to, among the built environments, the one that at least personally I'm most passionate about, which is the kitchen. So, I show you a kitchen, happens to be my kitchen, and I just want to remind you that this is a very familiar setting, and we can begin to ask, what is going on here? What microbial activities might be occurring that we might not know about that might actually be important for our sustenance? And so, of course, when we think about kitchen, the first thing that we think of course, is the products that come from the kitchen and the way that we actually eat it. And so we have to think about bringing it down to earth in the concept that we cook and we enjoy and share this food. But so much of the food that we consume actually has a microbial signature on it. And so there's so much microbial foods. And so bread and wine and cheese. And we might ask, why do we consume so many microbial foods? So I just ponder on that for a little while. And I'm going to offer you an idea. It's a crazy idea, but I think it's one worth pursuing in the future. And that is, I'm going to argue that which is known, that we've been doing this for a long time, and that there's a reason why we've been doing this for a long time as humans. And this is, how many of you guys have pets? Yeah, pets are not, you guys are not a group that have many pets. Anyways, maybe you have sometimes seen a pet like a dog. And it's out there, and the dog is out there lapping on the dirtiest puddle, right? And you say, oh, no, don't eat that. You have dogs and you tell them, no, don't eat that. You know, and the dog doesn't care. The dog eats that, and the dog eats all sorts of other stuff. And does he get sick? No. But if we were to eat that stuff, we would get sick. So I think there's a reason for that, because I think for evolutionary reasons, and this is the hypothesis of a colleague at Harvard, in this book, wonderful book by Richard Wrangham, Catching Fire, How Cooking Made Us Human. So the thesis is very, very compelling. So before there was Homo sapiens, Homo erectus was already catching fire and cooking. And the argument he makes is that that allowed humans to do things that no other primate could do. They could go out longer times because they could get much better caloric product out of the food because it was cooked. It was already partially degraded. And cooking made us human. But at the same time, that cooking reduced the microbial load of many of our foods. So all of a sudden, maybe not all of a sudden, along evolutionary history, maybe it became sort of dangerous to eat all that stuff, right? Like rotting animals and dirty water, because we were not exposed to all those organisms. So we needed to develop foods and beverages that were hygienic. You know, Pasteur said the healthiest and most hygienic of all beverages is wine. And there's a good reason for it, and I'll go into it. But it's clear that as humans have had civilization, they have been accompanied by microbial foods. So there is evidence, for example, as old as 9,000 years ago from China, that there's some pots that contain residues of alcoholic fermented beverages. So this has been part of culture since the times that culture started. So microbial foods are no mystery. And what do I mean by fermentations? So let me get into that very briefly, because it underlies the reason why microbial foods probably began to be drunk, because they were more hygienic and eaten. So normally, if we are respiring, like you are all doing right now, you're breathing, you're respiring, that means you are able to take the nutrients that you eat, such as sugars like glucose, and you can convert that into CO2 if there's oxygen around, and in the process you make energy. But when you stop breathing, try for a little while, then your pulse will go off, right? So it's not very good for us. But microbes are phenomenal, because they can stop respiring, many of them at least, and they can begin fermenting. And fermenting, we mean simply that they're going on with living in the absence of oxygen or any other thermal electron acceptor. And at that point, in the general case of microbial foods, we can go one of two routes. It's either alcoholic fermentation, and that releases carbon dioxide and ethanol, and that gives us wonderful things such as wine. But the other fermentation is lactic acid fermentation. And that gives us lactic acid, and the acid precipitates protein, gives us wonderful foods such as cheeses. Now, I lied when I said that we cannot ferment. We actually can do, but we cannot do it for very long. So if you have ever gone running for a long time, you reach your lactate threshold. You begin to accumulate lactate, so you can, for short times, actually carry on with lactic acid fermentation, but not for very long. Not for very long. But the key point really is that ethanol and lactic acid are such compounds that very few microbes can tolerate concentrations that are high. And therefore, wine becomes, by itself, by its own production, by the fermentation of grape juice, it prevents the formation of the growth of other microbes, and similarly with lactic acid, which means that this sort of foods were sustenance that probably were able to be used by early humans in a time when they were beginning to get less and less exposed to diversity of microbes. So knowing this, let's have a look at some of these wonderful microbial foods. So here, for example, we take some flour and we add some yeast, and we just let it rise because it's making the CO2, right? So this is the CO2 that is being evolved, and that's what gives us this wonderful bread. Then we bake it, we enjoy it, and it's a lovely microbial food. Now, if you just take, it's fascinating, because if you just take the same grain and you mash it up a little bit to release some of the sugars, and you wait a little bit longer, you get beer. So, and historically, it's fascinating, because beer and bread co-evolve, in the sense that many of the starters in the early Mesopotamian cultures were actually bred to start the beer to go. And so it's very, very well annotated that beer and bread were combined, just because they're basically the same thing, just one of it has a little bit more water, and one of them doesn't get baked. Now, this is all done by having some yeast. You can imagine that they really have yeast back then. We'll get back to that, because they didn't, at least not in pure form. So you can also have cheese, and wonderful cheeses. And the first thing that happens in making cheese is that you generate lactic acid, a bacterium, a lactic acid bacterium, such as lactobacillus, can just grow on the sugar, primarily lactose from the milk, and in thus generating lactic acid, that precipitates the protein, becomes the curds. That's fresh cheese. But fresh cheese, it's okay sometimes, but the really wonderful cheeses are the aged cheeses. So that's where microbial interesting things begin to happen during aging. And in sensu stricto, it's not fermentation, but we still call it a fermentation product in the vulgar language. It's okay. We say it's a fermented product. And I think it's very nice if we begin to look at what's happening in the rind. Because the rind really is a remarkable ecosystem of microbes that begin to grow, and the microbes that are growing give rise to other microbes that can feed on them, and on this ecosystem goes. And to provide us with our first demonstration of just how lively cheese rind is in terms of an ecosystem, Scott is going to provide us with some views live. This is a live demo of cheese rind.
Thanks, Roberto, and thanks also to Jane and everybody for coming out tonight. So I want to do a little exercise here to start out. We saw the pulse of the planet, we saw the bread rising, but I don't think we've seen any actual microbes yet. So we're going to use the microscope here. But what I want to do first is a little exercise. If I can get somebody to volunteer, out here and just look. This is a piece of cheddar cheese that I bought yesterday at a local cheese shop. And I just want somebody to look at it and tell me what you see here.
Flaky white crust on the outside.
Okay, flaky white crust on the outside. Okay, that's a good explanation. So that's what we can see with the naked eye is flaky white crust on the outside. But what we're going to do now is take it here to the microscope and put the light on. And here we're using actually a pretty low power magnification relative to what we can see with some other microscopes, but we'll just sort of start to explore this ecosystem of the cheese rind and see what we can find. So I'm going to move around. And what I'm looking for now is what I call a cave that occurs on the surface, and it might take me a minute to find what I'm looking for. All right, there's a couple.
There we go, there. Stop, focus.
So these are cheese mites, everyone. And this is a part of the cheese rind ecosystem. And so this particular cheddar cheese has a lot of these mites. It's a characteristic part of this cheese, actually. But you'll find these on any aged cheese in lower abundance. So even a blue cheese, you might find some of these guys hanging out. Looks like that guy's tugging him along there. So yeah, this is, sometimes they're really piled up on each other in these little caves. So this is our first demonstration, and I'll spare you guys the mites now. Just to show you this, going past the limitations of the human eye. So these are not quite microbes, but they're right at the edge of our human vision. And so we can't see them with the naked eye, but here at 40 times magnified, we can see this crazy zoo on the cheese rind. So what do you think about this, Roberto?
I absolutely love them. I must tell you that one of the reasons you like that crunchy, one of the reasons you love that lemony taste, that actually is true. They provide the lemony taste, lemony notes. So next time you go and taste cheese, lemony notes, remember. It's just part of the food chain. So they have their own food chain. First there's some bacteria there. They are degrading the remaining food that the cheese has, mostly lipids by that time. Those bacteria then allow for some fungi to also grow and begin to form a forest in there. And these guys, the mites, are basically the top of the food chain there until we come along and we eat them. But, so that's, anyways, a little fun demonstration of living organisms in the rind of some cheese. So you get the idea that these are wonderful microbial ecosystems, but they don't only exist in the surfaces of cheeses or in the, and we don't only encounter microbes in the foods, but basically everywhere else that we can see here in the kitchen, there are microbial ecosystems in the surfaces, in the people love to mention the sponge and the coffee machine. So everywhere, the fruit that is lying here, it's all coated with microbial ecosystems. And the question is, where do they come from? Where do these guys come from in the surface of the kitchen counter? And we think we're very, very clean, and we are. But the reality is that we as humans are ecosystems as well. So every person here is containing their own very individual microbial cloud. This is a fact that we've learned in recent years, is that we carry microbes on our surfaces. We're constantly shedding them. Every time I'm speaking, microbes are coming out. So the microbial cloud surrounds us constantly. And in fact, it's the drippings of that microbial cloud that populate houses. So they really are your microbes that are generating the microbial ecosystems that are sitting in these various locations. And to show this in a graphic way, I'm going to show you a video that was generated by, in fact, the Sloan-funded Center for the Built Environment, Microbiology of the Built Environment in Oregon, that used to be headed by Jessica Green. And so here we have the animation of microbes that are sitting on the surface of, in this case, a working surface of a designer. But the point is that we can ask where are them, and these various colors are meant to indicate the types of microbes. And you can see that the colors match with those microbes that are on the surface of the individual that works there. So wherever you work, wherever you want to look at your keyboard, your iPhone, you have populated those surfaces. It's not that those surfaces are dirty. I mean, they are as dirty as you are or as clean as you are. They are basically containing the microbes that you have. So let's go back then to this idea of the bread. I said you could make bread by adding some yeast, but when they were in Mesopotamia, there was no yeast to add. So what did people do? Because the reality is that the person has this microbial cloud. And that microbial cloud contains yeasts, and it contains lactic acid bacteria and many other organisms, many, many others. But when we take flour and we just wet it and mold it by hand, if we just wait long enough, it will rise the same, and it will make wonderful bread we call sourdough. And the reason we call it sourdough is because it doesn't have only yeast, but it has also the lactic acid bacteria, which provides that delicious sourness. And because both of these are growing, it tends to last a lot longer. So that's the reality is that there was sourdough a lot before there was really French baguette, etc. So this is something that we do all the time. And you may not believe that our hands are a wonderful ecosystem. So for that, we have our second demonstration, for which we would like a few volunteers.
The volunteers can start to assemble up here. So what we're going to do now is...
We're going to take 3, right?
Yeah, 3.
The first 3 to... Yeah, here, we have this. There we have 1, 2, 3. Okay. All right.
So this animation that Roberto showed is great. It shows us the microbial cloud. We know that they're there, but of course they're microscopic, so it's still a little bit abstract. So what I'm going to show you now is a little bit of a trick that we can do to see these microbes that are all over our body. So what I want you to do is this is totally sterile. There's no bacteria on here yet. What I want you to do is when I open this, just go ahead and put your handprint down on here and just gently let your hand rest on the surface, and then after a second you can remove it. Just make a nice handprint. Are you guys brothers?
Nope.
Okay.
We're not related.
All right, because we could do 2 probably on one, but so who wants to go first?
Okay.
All right, go ahead. And what we'll do is we'll get your
share the
email,
plate.
and
There
we'll
you go.
actually incubate these and send these to your parents afterwards. All right, that's good. You can remove it. Cool, perfect. That looks like a good one. All right, who's next? All right, there we go. Perfect. All right, good release.
And maybe they took to put there, since we have 2 more.
All right, so we can get 2 on here.
So that we can have 2.
Yep, so just see if you can put it towards the side. Perfect.
Excellent.
Yep, not too hard. All right, and then one more kind of over here. It's all right if it overlaps with the other one a little
Rub
bit.
it on the
It'll
rug,
be
it's good.
more artistic. Perfect.
Excellent.
All right, nice.
That's it.
Perfect.
Okay,
now,
so we will take your emails, and in 48 hours you will be sent the photograph of your handprint.
Thank you, guys.
So, because it actually takes a little bit of time. So our live demo has to have something that is a bit of a trick, as was already said, and that is it takes about 2 days, but it's amazing, about 2 days for the organisms that are lying on your hand as they are given these nutrients to become visible to the naked eye in the form of what we call colonies, where you have a single bacterium or a single fungal cell or archaeal cell that will be deposited there, and it will grow to become millions and millions of cells in a matter of 48 hours. So to demonstrate that, Scott already did this before. He had just washed his hands, I think, and this is 2 days worth of growth. Perfect hand for making sourdough bread. So if we look closely at this gives us a sense of the diversity of organisms that we have. These are what I was mentioning as colonies. They are the descendants of a single cell that was deposited. But some of these guys grew very fast, so they grew big. Some of them, you can see, many of them grew very little. And in fact, probably the vast majority didn't grow very well because that may not be its conditions. In fact, in many cases in the microbial world, we do not know how to cultivate many of the organisms that are out there yet because we just don't know enough about them. That's what's making the microbial world so exciting. It's a really universe to be a new universe to be discovered. So this is implying that there's many different types of microbes, but doesn't even begin to touch the magnitude of the diversity of microbes that are out there. And so for that, we will draw a little diagram, or attempt to draw a diagram, where the area that is presented in the diagram is related to the number of species that we think are out there of microbes in this planet. So in this microbial diversity, well, you cannot see the circle very much, but there's the outline of the circle. And this happens to be, in my computer, 3 inches. I don't know, let's say this is maybe 6 feet in diameter. That means it is about the radius 3 feet, pi times 9. You can figure out how many square that is. The important thing is that we think, as calculations go, and take this with a grain of salt, that there may be as many as 1 trillion microbial species on the planet. So those are the latest estimates. Now, why do I say take it with a grain of salt? Because we're still grappling with how to define a species. Actually, that happens at all levels. We're still grappling to define what really is a species. But we know that there's a lot. And when we compare that to plants and animals, where the current estimates are around 10 million, then we are thinking that the diameter of this circle, well, I exaggerated because I don't have a font that's small enough to give you the real size, because the real size has to be 10,000 times smaller in diameter, right? Because it's 100,000 times fewer species. And we have a wonderful diversity of species of plants and animals, but just the genetic diversity in the microbial world is unimaginable. So thus, it is not surprising that our homes would be actually full of microbial diversity, which is we call my microbial castle. That is, we might think my home is my castle, but the reality is it's my microbial castle. It's dominated by microbial life. And it is really there because we seeded it there. And thus far, I've mentioned foods, and I've mentioned the fact that we're coated with bacteria, and by and large, those are beneficial. And in fact, I would say that the big change in mentality that has happened, and you'll see it reflected in the exhibition, is that we used to fear microbes for good reason, because some of them are quite nasty, but the vast majority really are not pathogenic. In fact, many of them are beneficial, and most of them are actually quite important for maintaining our health. But there are some dark individuals lurking in our kitchen. And surprisingly, I will argue, they are in the place where you might thought they wouldn't be, because we have it there to prevent the microbes. And that's the refrigerator, right? And the reason we have refrigeration is because we want to preserve the food for longer, and we know that microbes grow slower at colder temperatures. But that means slower. It doesn't mean they don't grow. It means slower. But what happens, I don't know how many of you guys have refrigerators in your house, probably more than had pets. But I bet that you put your leftovers there, right? And then the leftovers go in the back. And then 5 or 6 weeks later, you say, I remember I have the leftovers. Why don't we reheat that spaghetti sauce, you know? That's a problem. So don't do it. So, because in fact, it is well known. And the problem is, for example, in milk or in apple cider. Let's take apple cider. It's wonderful, because apple cider, if you give it some yeast, fresh apple juice, you'll get nice fermented apple cider. It's quite nice. But now you put it very cold, then there's an organism that grows much better in the cold than yeast. For example, Listeria monocytogenes. Now, Listeria is not a terrible pathogen, but it's nasty enough that you don't want to be having it in your cup of apple juice. So please, the refrigerator, use with moderation. And clean it every now and then. Now let's think about other foods that you keep in the refrigerator, like your crispy lettuce, right? There's the crisper, it's called usually, and you open it and there's your nice lettuce and you take out, and it's true. Your lettuce will look nice on the plate and it's going to be quite crispy, and you think, oh, it's been triple washed with all sorts of stuff, and So what we can do the same thing as we did with the handprint. We can print the lettuce. And even though this was actually a print from cabbage, this is the kind of diversity that you can see. Wonderful diversity of microbes. And they're actually perfectly fine to eat. It's okay to eat your lettuce if it's nice and crispy and fresh. But I think it's quite nice to look at the lettuce and these organisms also as an ecosystem. So if we look at the leaf of lettuce, and we might ask what might be going on there. There's lots of bacteria, lots of other organisms. And there, again, you could easily find at the top of the food chain interesting predators. So Scott is going to show you some of the other things.
All right, so here we go again. I'm going to come over to the microscope. I promise you that if we can find the organism we're looking for this time, it will not be as creepy as the cheese mites. But it might take us a little bit longer to find one. Let's see.
So now we have, this is, for those of you who have read the Microbe Hunters, this is the Microbe Hunters of today. Scott goes into the microscope and begins to hunt. It's also like the 21st century voyage of the Beagle. Right? We're going out there to explore new worlds by going into the through the microscope.
That's what it feels like, yeah, definitely. Okay, so we're in luck here.
Hey, there.
So this is a tardigrade, or a water bear, as it's commonly known. And, well, you probably won't find these on most pieces of lettuce. It's completely plausible that they could have come in as a hitchhiker on the lettuce. This one here is a larger species of tardigrades, as tardigrades come. Typically, they're smaller.
Ooh, look at him. Look at his eyes.
This one also has a nice orange pigment, and you can see the eye spots there, and you can see the gait of the water bear, its motion, how it's kind of lumbering, and that is, in fact, how it's got its name as a water bear, as it's just here
Can
crawling.
you tell us about the dimension of one of these, Scott?
So these are actually big enough to just be seen with the naked eye, so they're kind of right at the edge of sight. Around up to 1 millimeter, but most tardigrades are a few hundred millionths of a meter, or even smaller, and parts of their life cycle are completely microscopic, such as their eggs. But you could really sit here and watch the tardigrade all day long, at least I could. So there's actually quite a few in here, and they're all interacting with each other and also eating other types of microscopic animals that are in the culture called rotifers. It looks like he's going to turn around maybe. We'll see. So some tardigrades are predators that eat other microanimals. Others are sort of vegetarians that eat algae. There's a huge diversity among these tardigrades, among the microbial world, hundreds of different species, and they're found everywhere. Yep.
Well, why don't you answer it?
Yeah, that's a good question. You know, certain people have different definitions for microbe. The two of us, we consider anything that's invisible to the naked eye as a microbe. So, you know, microbe as shorthand for microorganism. So these guys are sort of, like I said, similar to the cheese mites, right on the edge of what you would consider a microorganism.
I love them because, in fact, I believe that in this particular ecosystem, they're the top predator. But in fact, they are feeding on all the microbes that are really the standard size microbes in the microbe and around. But I think one thing that you will love seeing at the exhibition is we have a chart that talks about manifestations of microbes and larger animals, and they span the edge of sight. So, for example, microbes, you say, have to be unicellular. But we know, for example, the myxobacteria make fruiting bodies just like a mushroom, as it were. And you can see it, and it's part of their normal life cycle. So is that a microbe, or is that... So you know, the beauty of that, I think, is that nature has evolved this fantastic spectrum of sizes. And I am very happy with the name bacteria and the name archaea and the name eukarya. They are very important to define. But a microbe is a more difficult one to define. It's an operational thing you cannot see. It's microscopic. But anyways, that's, ooh, look at that. They got quite nice.
Yep. It's just exploring around.
Exploring around. So what happens as this little animal is foraging on what it feeds on. Those that it's eating are actually feeding on something else, and eventually that lettuce leaf that we were talking about actually gets, let's see, oh, you must bring me back. There, that lettuce leaf doesn't look so good anymore. It's been sitting in your refrigerator for too long, and it goes limp. Let it rot. You know, this is the process of rotting, which is a very wonderful way that nature has of recycling. It's just recycling organic matter, bringing it back into that other things can grow on. And so the only thing that we recommend is you don't let it rot in your garbage can, or you don't let it rot in your refrigerator. But if at all possible, you can compost it. And now in Cambridge, at least, we're fortunate enough that we have city-wide composting. So maybe some of you live outside where there can be some compost piles. And composting is a wonderful microbiological activity that eventually leads to a lot of growth of nematodes and other small animals, and eventually returns all of these things back to the soil. And so you might wonder how this stuff smells. And so yes, maybe at the beginning, depending if you put a lot of meat rest or fish in there, it's going to smell. Protein, when protein degrades, it doesn't smell too nice. But it's all part of going back into the earth and becoming soil again. So we have our last demonstration, and it has to do with olfaction. So we won't ask for volunteers, but what Scott will do is he has some of these microorganisms that are involved in making soil, and he will ask for the opinion of any of you who are brave enough to give it a whiff.
This is a culture plate containing some of the common bacteria that we find in the soil, completely harmless to people. And what I need is another volunteer who wants to take a little. You actually don't even need to open it. You can just crack it and just smell it a little bit like that. And tell me what you think it smells like, and then I'll tell everybody else. Yeah, just smell it through the edge and see if it reminds you of anything.
Yeah, smells like compost.
Like compost?
All right.
Okay. Does anybody else? Oh, right here. Okay. You didn't see it. All right, you can go too. All right. What do you think?
Smell that bad.
Yeah, it smells. I think it smells good too.
It smells good.
So what else might it smell like besides compost?
It smells kind of earthy.
Earthy?
Stop right there, stop right
that's
there.
That's good.
We got the right answer.
And then your turn. What do you think?
My answer is the same as his.
Okay, earth. Perfect. Thank you, guys.
So he said it right. It smells sort of earthy, because the smell of earth is due to a molecule which has the perfect name, geosmin. It means the smell of earth. Geosmin is produced by many soil microorganisms, in particular a family of microorganisms known as the actinobacteria, although some fungi also make it. And the actinobacteria are what we call the smell of earth. And what's fascinating about these actinobacteria is that they make molecules that have the properties to kill other organisms called antibiotics. We don't really know what antibiotics' role is in nature, although we have some examples, beautiful examples of where they are actually being used to selectively kill some organisms over others. But what's beautiful about geosmin is that the organisms don't make it at a time when they're growing, but they make it at a time when they have reached the end of their growth and they're making their spores and they need to disseminate. If you think about a mushroom, the mushroom comes up and it lets out all its spores, and that's for dissemination. And the air can take it. But these spores are not able to go with the earth that much. And so the hypothesis is that when little animals can smell geosmin, they can come and crawl all over these spores, get their feet full of their spores, and then go off into a different place and let the organism disseminate that way. Now, I love that because, you know, dogs, they have fantastic sense of smell, but we don't. We really have lost most of our sense of, we have fantastic good eyes, but we've lost our sense of smell, with one exception. We smell geosmin, something like 10,000 times, at lower concentration, 10,000 times that which anything else that we can smell. We have kept the capacity to smell geosmin through evolutionary history. And this is complete speculation. But I argue that for the spores, it's good to maintain geosmin because some little animal can smell it and it can disseminate it. But for us, it's good to smell geosmin because if we were back in the day when evolution was beginning to make us human, we were walking barefoot. And we could smell geosmin, and we could also get our feet full of these spores. And the beauty of it, that remember that these spores make antibiotics. And so they might be argued that that's a good way to get that ecosystem that might be your skin in balance. So I'm suggesting that when spring comes, and it will come, you consider taking a walk in the forest, smell the geosmin, and if you're brave, take your shoes off. So with that idea of this ecosystem and how we are part of a world that is surrounded by microbes, I just want to take you back to the very, very high view of the satellites, that we consider again just how fundamental the action of the microbes is and will continue to be in this planet to keep it alive. And that the best we can recognize that, and the best we can remain informed and learn more about them, the better it will be for the future of the planet. So thank you very much.